Today we start the physiology of the eye, and to understand it properly, we need to start with the favorite subject of all students, physics. Not just physics, optics. Don't worry, it's not. I just wanted to remind you what light is and which properties of light we have to consider when we talk about eye physiology. First of all, light is the wave. Do you agree with me? And wave can be characterized with a different length. As you can see, and light is electromagnetic wave. Okay? So it follows the same rules as other electromagnetic waves, same laws of physics. And you can see that the spectrum of electromagnetic waves is huge. The longest waves with the lowest frequency are radio waves, while the waves with the highest frequency, the shortest ones, are the gamma rays. And the humans can see in a very short range of spectrum, which is often called a visible spectrum. It spans from approximately 400 to 700 nanometers in wavelength, and it's flanked by infrared light, which has longer wavelength, and UV light, which has shorter wavelength. Does that make sense so far? It's waves. Waves can go through different media, through air, through water, through glass, diamonds, and whatnot. Does that make sense? Now, quite surprisingly, is that light is not only wave, it's also a particle. Huh? Yeah, it's photons, but it's called the de Broglie principle. There was a huge sort of a feud between Newton and who was the second guy? Huygens. Newton and Huygens. Newton thought that the light was wave, and Huygens thought that the light was photon. Well, particles. They didn't have a name of photon at the time, and they just couldn't come up to the conclusion. You know what's interesting about light? In some experiments, light behaves as the wave, and in some experiments, it behaves as a particle. So Louis de Broglie, French Viscount, is that, or Baroness, whatever, aristocrat and physicist, he suggested that light is actually both photon and a wave. And you will see how in our discussion we use both qualities of light. When we talk about the color and our perception of color, we talk of light as the wave. And then as we talk about physiology of transduction, we talk about it as the photons. So what we see is the light that is reflected from the object. Does that make sense? For instance, this table, which is black, means that very little light is reflected from the surface of this table. Most of it is absorbed. Does that make sense? For instance, I don't know, this blue can reflects blue light. So it absorbs the part of spectrum that is not blue light. So if this is blue, this part of the spectrum gets absorbed. Does that make sense? Just, to understand what we actually see. Now, we can see different wavelengths. We can see this whole spectrum because we have 3 different types of cones. We have blue cones, green cones, and red cones. They all get names for the color that they're mostly responsible sensing. But if you would look at each of them, first of all, the spectra the spectra that they are capable of sensing. Spectra overlap. The spectrum for the blue cones overlaps with the spectrum for the green cones. So these overlaps, okay, these overlaps allow us to see different colors. Does it make sense? That's why we can see from yellow to blue. You may ask, there are people that can see more than others. I don't know, how would you guess this, but there's a report, there's several reports on people who have fourth cone. It's known, it's not like, you know, something that you will read in the most trustworthy newspaper, National Enquirer. It's real deal. It's like real science, real anatomy. There are people that have fourth cone, and among those people is an artist, a woman that she makes like paintings, and your paintings have absolutely wild combinations of colors, not abnormal, just rich. And she describes her colors, and she sees more colors than we do. It is believed that she has an additional cone that she's able to use to see like more in the UV lights part of the spectrum, which is pretty wild. Okay, she sees more than we do. Now that's about the reflection and wavelengths. Next, about the refraction. It is important to understand how light changes its path as it gets in the eye. When light changes medium through which it goes, say from water to air and vice versa, it refracts. Okay? Light refracts when it crosses the boundary. Does it always refract when it crosses the medium? Well, in some cases, you probably will not be able to notice it. Window glass. Technically, you know, if you ever walked into the glass door, you can imagine that it can be squeaky clean and you will not notice it. There's no refraction. Does that make sense? You see what I'm saying? But if you would put like a transparent aquarium instead of the glass door, you will see that something is different on that other side. You will see the refraction of light. The image that the light that goes through water gets refracted significantly. The light that goes through the flat glass doesn't. The refraction coefficient of the medium is what shows you how strong the bending of rays is. Does that make sense? It is shown here. You see the light that crosses that glass of water refracts at the border of glass and water 2 times. And you see that distorted image, slightly distorted image of the straw. Does that make sense? And that refraction, that bending, also, you know, light as it goes through different media, actually the speed of light changes. That is partially why it refracts. Now, another thing interesting, so the material with the highest refractory power is, what do you think it is? It's diamond. Diamond has the highest refractory power of all transparent materials. It's incredibly high. That's why it's so shiny. And that's why professionals can tell diamond apart from, you know, non-diamond. Refractory power is different. So if we will translate that experience to convex lens, which are the lens in the human eye, we will see that the light that crosses the lens the light from the image that crosses the lens will lead to the fact that image on the other side of the lens will be rotated and upside down. Does that make sense? the image on the other side of the lens will be rotated and upside down. Look at me. What is actually on your retina is me upside down and left and right switched. So I move this hand, it's on the right side of the retina now. Does it make sense? Why you still see me normal? When do you learn to see everything as it is? You're a little baby. How? How do you perceive the world? What are sensations that allow you to explore the world? Somatic. Great, somatic. So you see something and you start to explore it when you're a baby. And initially it's a conflict, of course, because what you see is upside down, but what you feel is not. Eventually, brain processes those two seemingly opposite perceptions and put them together. It says, okay, so just flipped image, I'm going to flip it back in the thalamus. Does that make sense? So after processing, you see it normal, although the image on the retina is totally opposite. Does that make sense? Sorry. No. That's a brain processing. That's entirely brain processing that we learn as we babies. It's entirely brain processing.
So we just make that in different cameras.
Say it again.
So like the flip image and then reflipping it, we just do that with cameras.
Oh yeah.
And then you highlight the red, the green, and the blue as being so like we do that with screens.
I'm not. Now here's the thing. So I'm not. In screens we have red, green, blue. RGB system. I don't know what is the sensitivity of RGB system like regular TV. What are the spectra? So I'm not an engineer. But in the camera, at least we're aware that the image flips. So you can flip it back. Think about this. When you're a baby, you're not aware that the image is flipped. You think, oh, that's how it is. It's normal. And then brain starts to process that conflicting information and comes up with a solution. Okay, that's what it should look like. And automatically reflips it back in the visual cortex. Does that make sense? So lens that we're going to be dealing with, lens in the eye, are convex. Okay? We have to, and the convex lens, they bring focal point usually closer. The more bulged, the closer the focal point. Okay? So the more bulged the lens, the closer image you can see. Okay? The concave lens will make the image, the focal distance will extend. You'll be able to see farther. Does that make sense? Now, we're going to talk about the physiology of refraction in vision, and that's probably going to be it for today. So first, normal eye. We're going to talk about normal eye. What happens? So normally, our ciliary muscles are relaxed. Okay? Which means that the lens are flattened, because when they're relaxed, when muscles are relaxed, they pull on the ciliary ligaments, ciliary zonules, and flatten the lens. Does that make sense? So in this case, from the farther point, light gets almost in parallel rays, refracted, inverted, and here's you have real inverted image on the retina. The focal point of normal distant vision is about 6 meters. Okay? So for normal eye, you're going to see the perfect normal eye. You're going to see the perfectly, sharp image from about 6 meters away. So those reading tables in a optometrist office test if your eye is normal, about from 6 meters. So ciliary muscles are relaxed, lens are flat, and your pupil is dilated. Does that make sense? And that distant vision is under sympathetic control. If you want to see something close, how can you bring the focal point closer? You need to bulge the lens. Does that make sense? Bulging the lens. So you bulge the lens by contracting ciliary muscles. When ciliary muscles contract, they get narrower. That's the contraction of ciliary muscle. They get smaller. Ciliary zonules, tension in ciliary zonules decreases. As tension decreases, lens bulge, and they bulge just because they are elastic. As they bulge, the focal point becomes closer. Does that make sense? And again, you see the image on the retina. One thing that accompanies close view is constriction of the pupils. Here's the thing. Look at the close vision. Look at the light rays that come from the close object. They are diverging, they're not parallel. Does that make sense? Now, hypothetically, you can imagine the light ray that goes by the lens, not through the lens, but by the lens. So it's not refracted. Does that make sense? It will still be sensed on the retina. And wait, don't write it down yet. It can still be sensed on the retina, and it will totally distort the image. Does that make sense? To channel only the proper light rays, only the least divergent light rays, pupils constrict. They kind of filter out those more divergent light rays. Does it make sense? Go ahead. Fuzzy. I had this. When I got my eyes tested when I was a kid, I got atropine drops in the eye. Atropine is the inhibitor of ACH receptors. So it's parasympatholytic. So it dilates pupils immediately. And I looked like a tiny little heroin addict for about 2 weeks. The gigantic pupils. And another thing, ciliary muscles are paralyzed, so you can't really accommodate. I couldn't really see far, and when I tried to read, the letters were really fuzzy because of the dilated pupils and inability of lens to bulge. Does that make sense? Yes. Right, but atropine blocks ACH receptor and it parasympatholytic. So parasympathetic component in me was totally inhibited. Only sympathetic remained. Does that make sense? And that sympathetic tone dilated the pupils. Does that make sense? And that effect, and actually it was amazing because it was just one time, I think it was one time, dropped it, like 2 weeks. It was really, really strong, yes.
So, how was your vision at night, though?
I don't honestly, I do not remember. I remember that I was happy because I had like a lot of school excuses for like 2 weeks. No homework, stuff like that. Now, we'll get to that. Well, not so much LASIK, but I will tell you something about the eye thing. So, okay. Let me finish about the reading and close vision first. When you read close, first your ciliary muscles are contracted, right? So it's a continuous effort, and your eyeballs have to go medially. So you produce continuous effort on the medial rectus muscles. So essentially you produce continuous effort on at least two pairs of muscles, ciliary muscles and medial rectus muscles. That's why you get tired reading. Your eyes get tired. Now, there are two structures in the eye that actually refract the light. Lens, and what is the first structure? Not the sclera. Another part. Cornea. Cornea. Does that make sense? Can you see that? Cornea. Refract the eye. Well, refract the light, sorry, not the eye. Well, if we will try to picture the light rays going through the eye, what they're going to cross, tell me. What's this? Huh? No. This. Cornea. Then they go through what? Anterior chamber. Then what's this opening? What is the hole right here? The black one. Pupil. Then they go through the lens. Well, cross that small posterior chamber. Go through the lens and enter the posterior segment. And then they finally reach the retina. Does that make sense? Have an idea of which structures are crossed by the light. Now, when lenses become more bulky and less elastic with age, the ability to accommodate decreases. Does that make sense? And that inability to properly accommodate, to change the focal point, is called presbyopia, the condition. Everything with opia refers to eyes. Now, we're getting closer to the nearsightedness and farsightedness. So in nearsightedness, in myopia, the problem is that the eye is too long, anatomically long. So the lens focuses the image in front of the retina. Does that make sense? So those of you who are nearsighted have concave lens in your glasses, which move the focal point to the retina, move it farther. Does that make sense? Does it make sense? They diverge light a little bit before your cornea. Does it make sense? Yes, that helps to seeing further. Okay, now that's called an inability to see far. It's called myopia. I have that mild myopia. I know that. Okay, when something far is kind of fuzzy, you put this concave lens in front of your eye, light rays gets diverged, it moves the focal point back and focuses it on the retina. In hyperopia, which often happens to the elderly, in elderly it's going to be presbyopia, mostly because the light in the lens are unable to accommodate Genetically, it can be because of the eye being too short, the eyeball being too short. The distance between the anterior and posterior poles are too small. So the focal point is behind the retina. Does it make sense? So if you put convex lens in front of it, then focal point will be moved to the retina. Does that make sense? If you have magnifying glass at home, look at the shape of it. It's convex. Okay? Does that make sense? Now, astigmatism is the problem with curvature, all curvatures of either cornea or lens, when they are uneven. Not just, not flat enough or not bulged enough, when it's uneven. Does it make sense? You can correct it with lenses that will correct specific parts of the cornea or lens, the artificial lenses. And we get into the laser procedures. Now, remind me please, which two structures refract the light in the eye? The cornea and the lens. Which structure do you think has higher refractory power? No, it's cornea. Cornea refracts light more. Can we accommodate cornea? Can we change its shape voluntarily? Like right now, can I change the shape of my cornea? No. Unless I do surgery on myself, no. Does that make sense? It's sort of a constant refraction. So what you can do with a laser, you can, now I'm lost for English. When you oh crap, that's embarrassing. You can change the shape of cornea with laser, like taking layers, like shaving, huh? Say again? Well, you could just cut layers in a certain way, like you're making a sculpture. Carving, like you carve new shape of the cornea. Does that make sense? So, and that's kind of forever. But you don't need any glasses afterwards. And now we use laser. But I'm proud to say that the first eye surgeon that actually started to do that was Russian guy in Soviet times, in 80s, or even like late 70s. He developed a technique to do the corneal surgeries to carve new cornea, new shape, without laser. It was actual surgical instruments, tiny little scalpels, okay? And of course it was under anesthesia. I think it wasn't general. I think it was local anesthesia, but they kind of did something to the eyes, so the patient doesn't blink. And I still remember they kind of showed that on TV. They said that they received new scalpel, which blade was at the width of the blade was 200 angstroms. To give you an idea how small that is, the size of one atom is 1 angstrom. So it was 200 atoms wide. Huh? Oh, needle would be like the size of that classroom compared to that scalpel. Okay? Yeah, so they cost thousands of dollars. But he was very successful and he helped a lot of people to, get rid of glasses. Okay? But yeah, so most of those surgeries, they deal with cornea because the lens is just too deep. Does that make sense? Okay. I will